new insight into neurodegenerative proteopathiesis. Thank you for coming.
All right. Thanks. It's very nice to be here. I hope you really have been waiting a year for me to talk, because I'll really disappoint you. I am under the weather, and so I'm just running on half-empty tank here. But I'll do my best to try to get to the talk. I'm excited to be here because one thing I feel that I'm not good at is computational work in bioinformatics. And it's one thing that people like me who trained in sort of basic molecular biology and basic cell biochemistry feel that we need to ramp up. And so to interact with you is very exciting for me, and I look forward to being the first of many interactions as we go forward. One of the reasons that I think Brian wanted me to be here was that we have this new protein folding disease fast forward initiative. The grant that went in said establishing the Center for Protein Folding Disease. And the medical school loved it. But they said, you know what? Can you avoid that term center for a while? If you do really well, we'll turn it into a center after a year or so. And things are going very well. We have 55 faculty on board. We have research hubs. We have a series of resource library components. Santiago is running a in silico protein fold analysis module within that. And I think he's already gotten at least 7 customers in a couple months, which is great. So it's moving in the right direction. And we'd love to have more people interact with us in this interdisciplinary approach to protein folding diseases. I'm a neurologist. I see patients who have degenerative brain disease. And these 4 individuals, of course, all have succumbed or are succumbing to neurodegenerative disorders, which have a lot in common. Ronald Reagan with Alzheimer's, Lou Gehrig with motor neuron disease, ALS, Muhammad Ali with Parkinsonism, and Woody Guthrie with a purely genetic disorder, Huntington's disease. These are all protein folding diseases. They're all diseases in which there's an abnormal aggregation of distinct proteins that cause problems that we don't fully understand, but which are the focus really of the PFD initiative. Today I'm going to give you a little background on protein misfolding and aggregation, and then I will talk specifically about my own laboratory's work, which has primarily been on polyglutamine diseases, although we're increasingly working on amyloid precursor protein and ubiquitin-2 and other proteins that are implicated in frontotemporal dementia. And then the final point is, as a neurologist who deals with patients who are dying from these diseases, you know what? We don't know the mechanism, and I will spend the rest of my career trying to figure out the mechanism, but we don't know it. We do know that these are toxic proteins. We know the genes make something that's bad. So there are strategies to try to eliminate that. So I'll bring up some of the strategies we've been going after. So let's begin with this. This is something you all know. These different diseases like Alzheimer's, Creutzfeldt-Jakob disease, MSA, Parkinson's, Huntington's, frontotemporal dementia, ALS, they all have these different distinct aggregates that form in the brain that have specific proteins within them. These are some of the deposited proteins that occur here. These are all amyloidogenic proteins. They're all amyloid-like proteins. And of course, the characteristic being they're sort of beta sheet-rich structures that lead to highly ordered fibrils over time. People often ask me, why are these diseases age-related? I mean, you've got these darn proteins being made your whole life. Why is it that you don't see Huntington's disease until 45 or Alzheimer's until 65 or 70? And this is a purely speculative slide, but my view on this is that cells are constantly making proteins that go the wrong direction. It's a very crowded environment. There's misfolding, there's aggregation. The cells are working all the time to keep a clean house. And as we get older, as our cells get older, due to lots of different things, there's an increase, I think, in proteotoxic burden, shown here as on the y-axis as going up over time, and that this really, at some point, causes a problem to cells and networks of cells, particularly in the brain, but it happens in the eye, it happens in the ear, it happens in the heart, it happens in the pancreas. All these things can happen. And really, all you need is something to kind of push it a little earlier to cause a disease that manifests at 30 or 40. That might be a specific mutation like in the Huntington protein, or it might be a specific mutation that underlies frontotemporal dementia, or it might be a traumatic brain injury that can push you further down this way. Obviously, this is where we want to go. We want to delay this curve of proteotoxicity, and we still don't know exactly how to do that. There's a lot of speculation about that. My view of disease, very simple, is I come from sort of a genetic background, but I think that it's a genes-environment continuum. There's not a single degenerative brain disease that I see that doesn't have a little bit of both going on with them. And so on the one end, I would say would be the polyglutamine disorders, Huntington's disease is the one you all know, but there are 9 of them. And these are ones where you only get the disease if you have the disease mutation. So in one sense, it's a purely genetic disorder. However, we know that in families where there's lots of exercise, or in mice that are given the mutation, who exercise more, it seems to stall the disease a little bit. So there can be ways in which the environment in which one lives can alter the time course of that disease. It's a fully penetrant disease, and it's on the pure genetic end of the spectrum. On the other end would be something like traumatic brain injury. Okay, pro football dementia. We all hear about that a lot. Those individuals have developed dementia after they've had repetitive concussive injuries to the brain. But many people who had repetitive concussive injuries to the brain are not developing dementia. And almost certainly, even though that is an acquired problem, there are genetic factors that contribute to whether one will or will not develop a progressive degenerative process that was started by an environmental insult. And the big ones that I see in my clinic, Alzheimer's disease, frontotemporal dementia or frontotemporal lobar degeneration, Parkinson's disease dementia or diffuse Lewy body disease, these are smack in the middle. These have, all of them have a significant genetic component, but it goes both ways. And the weirdest one of all, of course, is prion disease, which is at both ends of the spectrum. You know about Kruger disease, where contaminated brains were eaten by tribes. You know about people who've had cadaveric transplants and who develop prion disease. You know about mad cow disease. So those are acquired forms of prion disease. But most people who ate contaminated beef in England did not get mad cow disease. You have to have a specific polymorphism, homozygosity, in order to be susceptible to developing prion disease. So even on the sort of acquired end of prion disease, you have to have specific homozygosity in a polymorphism in the prion disease. And of course, there are families that have familial prion disease, Creutzfeldt-Jakob disease, Gershman-Streusel-Schenker, where you have the mutation, you get disease. So that shows you that it can really run that spectrum. That informs a lot of the research that's going on out there. If you think about Alzheimer's disease, most Alzheimer's disease is late onset after age 65, but about 2 to 3% is early onset and is familial. And in those early onset families, there are dominant acting rare mutations in specific genes. And we know all of those mutations enhance the production or the accumulation of amyloid beta in the brain. In fact, the original case described by Lois Alzheimer in 1907, Adele H. Auguste D. She had a mutation in presenilin. So this is the original case, which we all think of as the index case, is in fact a familial genetic mutation form. But even the late onset individuals have genetic risk factors, and those have been discovered now. We know of at least 13 different well-validated genetic risk factors, the big one being apolipoprotein E. It's a huge genetic risk factor. Frontotemporal dementia is another one in that category, and the number of genes that now, when mutated, we know cause frontotemporal dementia is just exploding. These are shown relative to the relative frequency, so there are 3 that are big and important. This is the tau expressing gene, this is progranulin, this is this hexanucleotide expansion, has a beautiful mellifluous name, C9ORF72. All of these can cause disease. There are families, there are individuals who have sporadic disease, and we're just now finding the genetic variants, the risk factors that contribute to that. I'm just going to for a second mention TDP43 because it's a really good illustration of sort of the behavior of these proteins and how mutations can alter that. So this is an RNA binding protein. It is the most common accumulated protein in frontotemporal dementia. If you look at FTD brains, the most common thing you see is accumulation of TDP43. It's outside in the cytoplasm. It makes gigantic inclusions, or it makes dystrophic neurites with these tufted abnormalities. And you can have a mutation directly in TDP43 that drives that. Okay, you can have that. But all these other mutations too, ubiquitin-2, valine-containing protein, sequestosome, that's P62, that repeat expansion I mentioned, etc. They all, when mutated, can cause the accumulation of the TDP43 protein. And in the last 3 years, it's become very clear that it's not just FTD. If you look at aged brains, the most common thing that's abnormally accumulated is TDP43. If you look at Alzheimer's, between 26 and 55% of Alzheimer's brains have, in addition to plaques and tangles, TDP-43 accumulation. And if you look at chronic traumatic encephalopathy, that's the pro-football dementia, and all the various traumatic injuries that can cause late life dementia, TDP-43 is a classic abnormality there. Why is that? Why is TDP-43 sitting at that interface of all these diseases? Well, the current thinking is that it's one of many proteins that have prion-like domains. It is an RNA binding protein. It has a glycine-rich domain that by several different informatics packages, fold analyses, are suggested to be prion-like, have a tendency to become prionogenic. The mutations tend to be in that domain, and when those mutations are there, the computer algorithms predict that they're more prion-like. Indeed, the protein tends to be aggregation-prone. But what it indicates also is that in the absence of any mutations in TDP-43, that protein, as a harbinger of the problems in the cell, can tend to aggregate. Okay, it's what it does. I think these both are really beautiful papers. They're both worth reading. This is from Paul Taylor's group, but this one with James Shorter and Aaron Gittler is just a really nice paper about how RNA binding proteins often have prion-like domains. About 1% of the human proteome is predicted to have prion-like domains. Okay? So this is inherent in our brains. So what's going on? Yeah. Why that's there? It's a deep question. You know, it depends. It seems like a large number. And no one's actually experimentally validated. Those are predictions. No one's actually said how many of these are actually prion-like proteins. Obviously, the story in yeast where you have things like SUP35, these are clearly proteins that allow the yeast cell to respond to environmental states in order to essentially adapt, if you will. And so it may well be that certain proteins have that capacity in neurons and other cells, and it serves some vital purpose. If you take the I'll come back to this because I'll talk about the polyglutamine proteins, and I'll bring up sort of my thought about that. So, you know, proteins often fold correctly, but they don't always fold correctly. If there's a mutation or if it's a prion-like protein, there may be a tendency to adapt a conformer that leads to the production of oligomers and aggregates. These can, in a template-mediated way, propagate, and they can do it in a very distinctive way with specific isoforms or conformers, or they can actually sometimes interdigitate in a promiscuous way. And over time, what tends to see is highly ordered, larger fibrils that form the plaques or the tangles or the gigantic balls of polyglutamine protein. Of course, this is occurring in a cell that's very crowded with proteins, and proteins are complicated. So this is from, you've all seen this map. This is from one of Ulrich Hartel's reviews, and it's talking about sort of the energy landscape that newly folding protein sees in the cell. And remember, it's not doing in isolation. It's doing in a super crowded environment. And it may well be that the native state is achieved a lot of the time and is a nice low energy state and is relatively stable. But there are other states as well, including amorphous aggregates, oligomers, and really stable fibrils that can form. What I'm really showing this here for is to give you some sense that there is some capacity to move from isoform or conformer to conformer, and chaperones probably play a critical role in reducing those barriers for transitions. Chaperones are a key part of the machinery in cells that deals with these abnormal proteins. A very detailed picture of protein quality control in the brain, to me, has three big balls that are interconnected by arrows. We used to think of the proteasome and the chaperones and the autophagy as being kind of separate. They're so linked in so many ways. Chaperones work with the proteasome. Chaperones, in a ubiquitin-dependent way, can clear proteins. If proteins accumulate because the proteasome is not doing well, the autophagy system can take over. So these three critical elements of protein quality control are there all the time in cells and working hard. And chaperones play a role at almost every stage of protein production, protein handling, and protein clearance. And this is just simply from a Bill Balch paper where you can see that a transcription, as you translate a protein, even there, chaperones, this little pink ball, is involved at each of these points in going from a nascent chain to something that's properly folded, or disaggregating, or even aggregating, and clearing the proteins. When you clear a protein, it's usually through the ubiquitin proteasome pathway or through autophagy. the two other arms of the quality control. Now, my own laboratory has spent a lot of time on ubiquitin pathways over the last, I'd say that's where we now do most of our work, is in proteins that relate to the ubiquitin pathway. And I don't want to spend a lot of time on this cascade, but I think many of you know that ubiquitin is attached to proteins as a monoubiquitination or as a polyubiquitination that can either sort them to different parts of the cell or can clear them through the proteasome or other mechanisms. And you take a ubiquitin and through the cascade of E1 enzyme, E2 conjugating enzymes, and E3 ligases, you actually can add the ubiquitin on the proteins. And in the same way that phosphatases are the yin and yang to kinases, de-ubiquitinating enzymes or de-ubiquitinases or DUBs are able to take chains apart and take ubiquitin off of proteins. My own lab has worked on a number of these. We have some really interesting results I'm not going to talk about this E2 conjugating enzyme that is the only one we know of that instead of ubiquitinating lysine residues, ubiquitinates the amino termini of proteins. And we have no idea why we even have that E2. And that's something we're trying to figure out. We've made a knockout. The knockout tends to die at birth. We don't know why. This is very much a work in progress. In 2 years, we might have some sense about that. I will talk a little bit about some work that we've been doing on a quality control E3 ligase, and I won't talk about our work on FBX2, which is a fascinating E3 ligase adapter subunit that binds sugars on proteins, specific high mannose glycans, and so it's very important in the glycoprotein pathway. And I will talk about ataxin-3, which is a polyglutamine disease protein and is one of these DUBs. I will not talk about ubiquitin-2, our new favorite protein, which is fascinating, that actually is involved in clearance, probably both in facilitating chaperone function and in facilitating delivery to the proteasome. It's a well-defined cascade. Most of these will ubiquitinate lysine residues. Any kind of lysine residue can be hit. It has to be surface accessible typically. Again, this UB2W is the one that's hitting the amino termini. Because of the structure of that protein, it is unable to really see lysine. Those are just two papers that you might want to look at from our lab on that. Okay, so let's talk about the polyglutamine. Any questions at this point? Okay. Let's talk a little about the polyglutamine proteins. These are 9 different diseases. One of them is Huntington's disease, which Woody Guthrie had. One is a form of motor neuron disease called spinobulbar muscular atrophy. Andy Lieberman here works on that. He's really well known for that work. Bill Pratt's worked on this too. I think he's in the audience maybe. SCA3 is one of the hereditary ataxias, spinocerebellar ataxia type 3. These are very different diseases. In the clinic, we can easily distinguish them. Yet, they have the same basic problem, which is that there's an expansion of a polyglutamine stretch that we all normally have. We all have a polymorphic polyglutamine stretch in these proteins. But when the CAG repeat becomes expanded in the disease gene, it now encodes a much longer stretch of glutamine in the proteins. This is occurring in all 9 of these disorders. And you can model them any way you want, whether you look at a human disease brain, whether you look at a cell line, whether you make a mouse model, whether you make a fly model, worm model, whatever you make. If you go beyond the threshold into the expanded range, you start to see aggregation of the protein. You always see that. Big controversy in the field. Are the big balls of aggregated protein, are they bad or not? I don't really care. What I do care about is that that process is going on, and that is clearly fitting with the threshold for a repeat disease length. So somehow the problem of abnormal protein conformation, abnormal protein interactions, maybe it's merely an epiphenomenon that we're seeing the large inclusions, but somehow those are linked to the toxicity that exists in the brain. What's going on? It's back to your question, Brian. Why do we even have polymorphic polyglutamine stretches? Why do we have prion domains? Most of the proteins that are polyglutamine proteins are not hardcore scaffold proteins or enzymes. They actually tend to be modular components of macromolecular complexes. And most of them actually work in more than one complex. And it may be that there has to be a certain degree of flexibility in domains that allow them to be in this complex versus that complex. So having a polyglutamine stretch that allows for some degree of flexibility, some normal dynamic behavior of the polypeptide may be important. And what happens when you expand that? Well, we don't really know, and there's no crystal structure of an expanded polyglutamine domain. But the best view out there right now is that somehow when you have go from a normal length repeat that may have the capacity to take different shapes, you get a longer repeat, you may well have an intramolecular interaction that isn't an energy minimum and hard to get out of, and makes this very accessible and locks this into something that's not dynamic. And now you can start to get intermolecular interactions, and if there's a cleavage event that releases the polyglutamine domain from the rest of the protein, now you can really drive that completely. So the thought here is that you're losing that normal dynamic interchange. You have an increased propensity to aggregate, and if you have a cleavage event, that may actually cause more aggregation. And so polyglutamine diseases, many labs have shown that all three arms of protein quality control seem to be vitally important in handling it. And it was a number of years ago that after we had done some work on the proteasome and we'd done a lot of work on the chaperones, we realized there's a lot of crosstalk here and we need to be thinking about that. And a specific E3 ligase known as CHIP, or C-terminus of HSC7 interacting protein, was one that we went after because it links the chaperone pathways to protein clearance through the proteasome. And so we wanted to ask the question, does CHIP actually modulate these proteins in any way? I won't bore you with this long published experiments, but basically if we express wild type CHIP in cells, we can markedly reduce the amount of inclusions that are present there. This particular E3 ligase interacts with chaperones through this domain. And if you eliminate that domain, you lose that ability to reduce the inclusions that form in cells. So this is probably occurring in a chaperone-dependent, and we know also that it's occurring in a ubiquitin-dependent manner. We did this in zebrafish models as well, and in fact, if you add it in there, you can suppress toxicity in zebrafish. We tested this in multiple mouse models. I'm just going to show you from one, which is a transgenic mouse that expresses the SCA3 disease protein. And in this mouse, the disease protein is there. We can see it aggregating, but it doesn't kill the mouse normally. If, however, you reduce the amount of CHIP and eventually eliminate CHIP from the mouse, and by the way, mice without CHIP live, and they can live a full length of life. They die fairly rapidly if they have the mutant ataxin-3 protein, the SCA3 disease gene there. And if you actually, I won't show you the details, but if you look at these mice that have half the normal amount of CHIP, those mice become sick over time. They don't die early, but they become very clumsy on various tests, and they have more aggregation that occurs there. The key question for us was, is this phenotype correlate with some measure of the aggregation tendency? We wanted to see about that. And so we actually looked at these mice at early stages when they don't yet have large inclusions there. We wanted to see, is there evidence of biochemically detectable aggregated protein? And sure enough, there is. This is a native gel where you're seeing very large complexes. We're staining for the disease protein, so black represents the disease protein. These are non-transgenic mice, so naturally there's no of that disease protein. This is a mouse that expresses the mutant disease protein, but with normal amount of CHIP, half the amount of CHIP, and no CHIP. These are what we call microaggregates because they're not in gigantic inclusions. They are clearly complexes, somewhere between 700 kilodaltons and 1 million daltons in size. And they are resistant to STS, okay? So they are some kind of an early stage aggregate phenotype that's occurring there. We don't know that the microaggregates are toxic, but there's a correlation between the toxicity in the mice and the presence of these, and that's about as close as we can get. So what is CHIP doing in the context of that earlier diagram I showed you of proteotoxicity over time? Well, I think that what's happening is if you reduce CHIP, you're pushing yourself down on this curve, and I bet if you increase CHIP or something similar to CHIP, you could push yourself further out the other way. CHIP is not a good druggable target, but the chaperones with which it interacts are very druggable. You know that Andy's working on that. You know Jason Koswicki, who was here now at UCSF, has worked a lot on modulating HSP70, HSC70 activity, et cetera. So in that, what I showed you earlier about how chaperones play such an important role in so many spots, what's CHIP doing? CHIP is facilitating that degradation in a ubiquitin proteasome pathway. So let's come back to our diseases. same problem, expanded polyglutamine, and yet very different diseases. Why is that? Well, it really has to do with protein context. Okay, so it's not just polyglutamine aggregation per se. It is misbehaving of polyglutamine within specific polypeptides that have specific functions. And this is actually, I have to say, this is still a good review. It's old, but it was a good review. And if you want to read a little bit about it, and we realize there's no good reviews recently, so we got to go back and rewrite this. This just shows you the different disease proteins, what their functions are, what their sizes are, high resolution structures of the proteins here. And they have different sequences. There are different colors because they really don't share anything. And the yellow line is simply showing you where the polyglutamine stretch is. The one I started working on years ago when I was a postdoc was ataxin-3, in part because I saw patients in the clinic, but more importantly, in part because look how big it is compared to, say, that one. We could easily make the recombinant protein. We could play with it in a test tube. It was very nice. So a huge amount of work going on SCA3 disease protein, ataxin-3. It's a fascinating protein. It's a DUB. It's a de-ubiquitinase enzyme. It has a globular amino terminal domain that's highly conserved across phyla. It has a flexible C-terminal domain that's less conserved, that has several ubiquitin interacting motifs, and has the polyglutamine domain here. There are some similar proteins that we also work on that only have this domain that don't have that domain. And what this domain seems to do is it essentially gates the kinds of ubiquitin chains that this DUB will actually cleave. It's fascinating also because it's a pretty sluggish enzyme, but it often is monoubiquitinated very near the cleavage site. And that, for reasons that we still don't quite know, enhances the activity of the enzyme markedly. So maybe it is a positive reinforcement in the cells. If you've got a lot of ubiquitin around, this thing may get more monoubiquitin in the circumstances, and that enhances its activity. It can work on different kinds of chains, but we still don't know exactly what it does. Our best guess right now is that it works together with E3 ligases to edit the kinds of chains that are made when you ubiquitinate a substrate. It's been linked to ER-associated degradation, to agrosome production, functions in the nucleus, including regulation of transcription, and even autophagy, which I think is the weakest link here. But there's decent papers published by many different groups that says that ataxin-3 can play a role in all these. And when we years ago, together with Nancy Bonini, expressed this normal protein in flies, it was an incredibly powerful suppressor of various kinds of neurodegenerative disease protein toxicity. The flies are easy. Testing that in mice is much harder, and Lee Tsong in my laboratory actually crossed HD knock-in mice with ataxin-3 knockout mice, which are happy and healthy. They just have more polyubiquitin in the brains. And she asked the question, does it actually accelerate the phenotype? And a huge amount of work, and the answer is not really. So elegantly done. It's nice paper. You'll never read it. It doesn't make a difference. What we know is that flies and mice are not the same. We still think it's a very important protein, but it clearly is not critical for handling Huntington in the mouse model context. One thing we've discovered over time is that there's just a boatload of splice isoforms that are made. I mean, this is a problem that we're all experiencing. There are over 50 different splice isoforms that have been described for the protein. We've done a lot of work to try to say which are the most abundant ones, and there are about 3 or 4 that actually are reasonably abundant. And there's a really interesting 2 different isoforms that differ in that this one lacks the final exon, exon 11. This one has it. So you can see that this one has another UIM and this one does not. This is a more hydrophobic sequence. I'm showing you this because it becomes relevant in what I'm going to talk about next. Yeah. So this is what's happening. Normally, if you got the 3 isoform, you have appropriate splicing between exon 10 and exon 11, and you make the normal, most abundant C terminus of the protein. And this is the protein we tend to work with in test tubes and cells. If you get read through into the intron here, you get this end of the protein instead. So why am I telling you that? Well, we made a knock-in mouse model. No one made a knock-in mouse model. We really wanted to make one because it's the most genetically precise model in which to really study the behavior of the protein. And we made the model. No need to go through that in any detail. The knock-in has lots of aggregates, and it has a tendency for the protein to accumulate in the nucleus. We know this occurs in human disease. This is in the hippocampus. You can see that there's lots of aggregated disease protein here and those little red dots that you don't see in the wild-type mice. So it recapitulates at least some of the biochemical features of disease. And one of the first things we did in characterizing it, we said, we think this protein plays a role somehow in the nucleus, and we're worried that maybe we're wondering whether in disease states there's an alteration of transcriptional regulation. So we did RNA-seq analysis on discrete neurons in the deep cerebellar nucleus, which we know is a target disease, before there was aggregation in these mice. And I would say still a work in progress, but the most interesting thing we found is that when you had the CAG expansion in exon 10, whereas in wild type, you don't get much read-through into intron 10. In the expanded allele knock-in, you get a lot of read-through into intron 10. Okay? We actually were able to amplify this, and so this is what you get. If you actually get this read-through, you're going to get that alternative isoform that has a more hydrophobic end. We actually showed that in the mutant versus wild type, indeed, this part of the read-through has a polyadenylation sequence, and we actually sequence the RT-PCR product, it's polyadenylated. So we have the transcript that's the read-through, and we have evidence now that the protein is made. And when we look at this in the knock-in mice or in different transgenic mice or even in disease human fibroblasts now, we see that there's a much increased read-through into intron 10, somehow tied to the expansion of the CAG repeat there. This is interesting because the same phenomenon was described just last year in Huntington's disease. They have an intron read-through when they have expansion in the CAG repeat. So we don't know why, but somehow having a CAG expansion is altering the way these things are being spliced. And what it's producing is a different isoform at higher levels. And our data thus far suggests that different isoform 2-isoform isoform, is intrinsically more aggregation prone. And so that now becomes perhaps a seed for aggregation in these diseases. When we play with this protein, even with the expansion, it's much less likely to aggregate. So going back to that bulge diagram, in this disease, there's a huge issue with impairment in proteostasis. But as it often happens in science, it gets more complicated. We're now wondering whether there's actually a change somehow even in the RNA end of things. And there's so much more data in the last 2 years in neurodegenerative diseases that changes in tRNAs or in splicing are contributing to degenerative disease. And it may even be in this disease, which we think of as a proteotoxicity disease, that there's an alteration in the splicing regulation that's leading to a bad protein. So even in this classic protein toxicity disease, we may have some problems in impaired ribostasis. By the way, in other FTDs like that C9 hexanucleotide expansion, there's lots of data that this is impaired, and there's lots of data that this is impaired. And there's even the production of these really bizarre translated products out of the repeat. You don't need a start codon for methionine-initiated protein translation. You can have a repeat-associated non-methionine-initiated translation. These are called RAND peptides. And so you get all these different RAND peptides produced from these repeats. And these RAND peptides tend to be aggregation-prone. We're studying those in the laboratory right now with Magda Ivanova, who was the first recruit in the Protein Folding Disease Initiative. All right, how much time do I have? 15 minutes? A little therapy? Okay. All right, okay, all right. Okay. So the problem in this disease, of course, is the mutation at the DNA level that makes a transcript that makes a bad protein. And so all this stuff, a bunch of arrows, we're kind of faking our way. We're trying to figure out what's really going on. But we have a feeling that this is a problem. So if we can reduce levels of this, that would be good. And so one of the approaches we've taken and done this now for many years is to use nucleotide-based suppression strategies. RNA interference, antisense oligos to knock down the gene. And that's worked very well. And I'll talk a little bit about that. We've more recently come up with nice cell-based screens that allow us to screen for drugs that reduce steady-state levels of the mutant protein. And we're moving some of those into mouse models now to see if they work. And we've even developed a protein fragment complementation assay that's a little more difficult, but it's still pretty good, that hopefully will tell us about drugs that can block the oligomerization step. And we don't know that that would be a good thing, but if we can find those drugs, we can then test that. So a little bit about that first thing, RNA interference. RNA interference, you all know, is a way you can basically, in a very gene-selective way, turn off a specific gene and that makes a protein. And in the case of these diseases, we would love to turn off the mutant allele and eliminate the production of a toxic protein. And we've done that. It's a whole course in and of itself to talk about non-coding RNA regulation and the role of, RNA interference normally in biology. But what we do is we put a virus in that encodes a microRNA-like antisense structure or short hairpin RNA. It gets produced, it goes out, it gets made, and if it's against the ataxin-3 transcript, it can actually lead to cleavage of the transcript. We've done this in a lot of different ways, but in the studies I'm going to talk to you here, we actually target a specific region, the 3' UTR. Again, we used a microRNA-like design, so it was more biological. No, going into the brain, right into the brain. Well, we started all in cell lines, a lot of work. Then we go into the brain. So in this disease, the cerebellum is really badly affected. And so you can inject into the deep cerebellar nuclei, and it'll go up into all the parts of the cerebellum, and you might be able to actually stop expression of the mutant protein. And this is just simply the design of the experiment. We injected them early when they would have early symptoms, and then we looked at them 10 weeks later. We let some mice go to end stage. These mice tend to die at about a year of age. This just shows you the virus encodes GFP, so you can see where it's transduced. And in fact, if we inject down here, we can hit most of the cerebellum, and we get most Purkinje cells. That's that sort of workhorse cell in the cerebellum. We can hit those. And this is simply showing you a sort of low-power view stained against the disease protein. So these are different lobules of the cerebellum. This little strip of brown you see are the Purkinje cells. And this is a mouse that was actually injected here, and you had knockdown in this region. So if you actually blow it up, you can compare the level of ataxin-3 staining here versus here. And there's a good suppression in that particular model, that particular mouse. We can look at these by Western blot. I'll just have you focus here. These are sort of vehicle controls. This is the mutant protein. These are ones that receive the virus that would turn off the mutant protein. You can see there is a reduction. We knock it down by about 50%. Remember, we're not hitting the whole cerebellum. We're not hitting every cell in the cerebellum. So we think a 50% reduction is about as good as we can get in this situation. That seems to be the average. Key question is, we gave it. These are expressed for a long time. It's silenced for the life of the animal. Did it stop the symptoms of disease in these mice? Did it prevent them from dying? And the answer is no, it did not. I mean, this was very depressing. We spent a huge amount of effort into actually phenotyping these mice, determining the right behavioral things to look at. And then what we found is that instead of having our appropriately experimentally treated mice having appropriate studies that similar to wild type, they were no different. So we saw no difference in their ability in coordination tasks. We saw no difference in the survival of the mice. So what does this mean? Well, firstly, it was successful. We turned it off. We turned it off for the life of the mice. There was no evidence of inflammatory changes. The cells were there. They weren't dying. So it worked in that sense. But it didn't work in the sense that we didn't stop disease features. And it makes us think that the most likely possibility is that we've hit the cerebellum. But this disease affects the brainstem, it affects the spinal cord, it affects the basal ganglia. We haven't hit all those areas. That's something we're now revisiting with a different approach. Now, RNAi has been around for years. It won a Nobel Prize many years ago. 8 years after it was discovered, RNAi won a Nobel Prize. 8 years beyond from that, and it still hasn't led to a therapy. It may never lead to a therapy, but it is moving in that direction. We're at this point now here. We've done nice tests in mice. Some of these diseases, people are looking at monkeys. We haven't done that yet. I'm actually much more excited right now about antisense oligos because I can distribute them throughout the brain, and we're looking at those right now together with a company. Let's look here a little bit about this. So in order to find drugs that reduce steady-state levels of ataxin-3, Carmo made cell lines that expressed the ataxin-3 protein either with normal or expand repeat fused to luciferase. So it's a simple luminescence assay. Levels of luminescence tell you the levels of the protein. And She screened initially just the small microsource collection. She's expanded beyond this, and we're just about now to set up for high throughput screening. Out of this, she identified 10 molecules that survived that did a pretty good job of knocking down the protein. This is just an example of one of those, or 6 of those. And what you see in the blue is the change in luminescence. Here's control, and some of them work pretty well. The green is simply viability, so they weren't toxic to the cells. This is simply showing you that there's a dose response for one of these molecules that happens to be a cannabinoid receptor antagonist. Then we developed secondary assays. We made slice cultures, and we sprinkled the drugs on the slice cultures. This is tedious. This is a lot of work, but we're not going to do a full mouse trial until we have some evidence that a mouse that expresses the human disease protein, expressed from the human disease gene, has a reduction with these compounds. And doing this, we've identified 2 compounds that actually seem to reduce levels of ataxin-3, and those compounds are moving forward right now into the mouse trial. That's a lot of work. You have to do it in a blinded way, and most mouse studies, as you probably read in science journals and Nature, most studies that people do that show an effect in mice are never reproduced. They're just not replicable. So we have to be very careful about how we do that. And finally, using protein fragment complementation to actually try to find the early steps in oligomerization. It's very much like you use 2 hybrid approach. You take one fragment of, in this case, a yellow fluorescent protein, the other half of yellow fluorescent protein, neither of which by themselves is fluorescent. If you have a bait and a prey, in this case the bait and prey are both the polyglutamine protein. If they come together and they reconstitute the fluorophore, you can actually see luminescence or fluorescence in the cell. We've done this with fluorescence, but we have better results with luminescence using luciferase, so we've gone back and reconstructed these. We're still a work in progress. I'm not totally happy with our signal-to-noise ratio, but we're getting closer. When we generate a series of cell lines that express polyglutamine protein attached to fragments of, in this case, YFP, you can see that with the full-length ataxin-3, the mutant ataxin-3, a fragment that's highly aggregation-prone, you start to get these signals that we can easily see compared to some of the control ones. And this is in the luminescence assay where we've put fragments of luciferase in there. Again, we know that wild-type ataxin-3 is prone to form dimers anyway, so it doesn't surprise us. And we've seen increase with mutant in all of our cell lines here. And this is before you see like aggregates in the cell. So we think we are capturing early dimerization or oligomerization events. And in using this, this is simply a proof of principle test at this point, using a compound that we know reduces levels of aggregated ataxin-3. This particular compound is an inhibitor of HSP90, and it actually can kind of boost a stress response in cells. And with different doses of this drug, you can reduce the level of luciferase activity or luminescence that shows there. This is simply a control for viability. This is a different cell in which we have full luciferase. We don't have the fragment study, so we're not actually looking at dimerization per se, and there's no effect there with that. If you actually look at these particular samples, using that agarose gel approach I talked about earlier, where you can see the microaggregates, we actually see a reduction in that over time. There still is the protein there, but the aggregates are down. So we're excited about this, and we're hoping this can give us some clues to the role of aggregates in disease if we find compounds that can regulate it, and maybe some potential routes to therapy. And finally, and this is really important, no matter what we do here, we know that the cerebellum, which is an important part of this, has aberrant electrophysiology. So one of the things our laboratory has done, and now Vikram Shakate is his own faculty here on Faculty of Neurology, there's marked changes in electrophysiology of the Purkinje cells. And we've identified drugs that can actually modulate the channels and improve that activity and improve symptoms in the mice. So we need to be thinking about symptomatic therapy as well in these diseases. So that's kind of a whirlwind of where we're going. This is my lab as of last year. The individuals who did most of this work are Karma, who's hiding back here. Gotham was doing a lot of work on the protein fragment complementation with Masatada. Lee did the work on Huntington's disease that was kind of a bust in a way. And Graham done beautiful work on APP and FBX2. Matt discovered the UB2W protein. And Bishwa made the knock-in mouse. I'm very fortunate to be here. This is a great university. It was a fantastic move. I loved Iowa, but it was a fantastic move to come here. And I'm looking forward to the next 15 years of my career, collaborating with people like you. Thank you very much.
Thank you for the nice talk. So, any question?
Yeah, okay. I'm just wondering if you could tell us a little bit now about the protein misfolding.
Yeah, okay, so the Protein Folding Disease Initiative received $9.3 million from the medical school to create these new resources. It has 4 research hubs where we bring scientists together. The idea behind those research hubs is scientists from different departments with different perspectives can actually kind of bounce ideas off. It's not a lot of money, but it's a chance, enough money to start new ideas. Those research hubs include one that's focused on chaperones and their role in protein pathways. One is focused on the ER stress and modulating that. A third one is focused on vesicular events in neurodegenerative disease. And the fourth is looking at the relationship between metabolic disorders and Alzheimer's disease. So in all cases, they're looking at things that relate to protein folding and how other pathways affect that. The resource library has a lot of elements, including a newly revamped post-translational modification score. We got money for a new mass spec. Just came in last week. It's an Arbitrap, and it's going to be much better than what we currently have. Kojo Ellen Tova Johnson runs that. Santiago's about to leave, and can you say one sentence about your module before you leave and how that could relate to this?
So what we're doing is essentially we're offering services of protein informatics for analyzing aggregation propensities and protein disorder. So we're running on behalf of people who want to calculate protein structures. So different algorithms for making predictions of protein structure. Yeah. And in addition to that, so we do analysis of chemical kinetic experiments and signaling pathway experiments that are of signaling and pathways which are related to protein folding diseases, like unfolded protein response.
Yeah, well, and for what I'm hearing, people are really getting a lot out of that. There's also a mouse disease model compendium. There's a brain bank. There's a lot of different things that are there. The only informatics component is really what Santiago's doing. So we're excited about it. It will succeed if people use it, interact. And frankly, it'll succeed if we can get a donor who says this is really cool and will sustain it beyond the 5 years that they've been given to us. Already 4 grants have been submitted that list the Protein Folding Disease Initiative. We have boilerplate that can help with that. So is there any way that you, as computational biologists, as informaticians, would benefit from that? Feel free to contact me. We could definitely do that. Is that sufficient, Brian?
No, that's great.
Okay.
You know, John obviously has a goal.
Yeah, yeah.
Protein folding manipulation.
Well, we would love that.
Yeah, I'd like to go back to the beginning of your talk where you were talking about the basis for late onset. And I remember when people started to talk about this years ago that they were talking about the possibility that the stress response, or at least the function of chaperones, was deteriorating with age. And we actually tested that in a physiologically sound model for HSP90 and HSP70 anyway, and found that old versus young didn't seem to make any difference. It was quite competent. Do you know if there's any evidence for that model?
So there's, you know, if you look at the disease models that, and so I would throw out anything that's looking at human disease tissue, because human disease tissue from controls versus disease are like apples and oranges. And so you've lost cells, you have different cells that are there. So if you're trying to compare a specific protein in the control or age matched versus the disease, I don't know what it really means. So, but it's in the disease models, mouse models, where people looked at this. And there's pretty good data, at least in the polyglutamine diseases, that over time, before there's lots of degeneration of neurons, there is a reduction in HSP70. In Huntington's disease, we've seen it in Huntington's knock-in too. We looked at that in that paper that I mentioned. It was a negative paper. There was a reduction in Huntington, or HSP70. But no one's really done a careful assessment of whether, in fact, there is a reduction in the robustness of various stress responses, whether it's chaperones, whether it's the unfolded protein response, et cetera. My guess is that it's probably more nefarious and multifactorial, involving everything from changes in splicing and transcription and co-translational control to problems with appropriate clearance. But no one's really nailed that down. The other option for what's going on is that actually there's just an accumulation of protein over time. There's no change in the cell's ability to handle it, but there's just always going to be some accumulation. You can't keep up, and it accumulates over time, and the accumulated protein causes problems. I think that's less likely to be the case. There's no great evidence that the large visible aggregates under the microscope are contributing to toxicity. And the data in cell models of Huntington's disease show, but this is cell model massively overexpressed in the protein, is that the neurons that have diffuse mutant Huntington are more likely to die than the neurons that end up with large inclusions. And that kind of makes sense to me. I agree. I think that probably an oligomer or a monomer with a highly reactive polyglutamine surface is going to be more dangerous than a bunch of stuff that's been compressed into a structure that may well be walled off by factors that keep it sequestered. So those inclusions that we see are not simply the disease protein, they're probably also the cell's response to the disease protein and walling it off in some way.
More questions?
Yeah. Still, so the question was, is the altered physiology due to new channels or alteration in the channels that are already there?
Yes.
So it's still a work in progress. What we do know is the Purkinje cells are always intrinsically firing. They're a pacemaker cell, like a cardiac cell. And what happens is you get depolarization block in these particular Purkinje cells. And we don't yet know what channel is changed to make that different. We do know that if you actually enhance calcium-activated potassium channels, you can correct it, the depolarization block, but it hasn't yet been worked out. And what we'd like to think is that the same kind of, because that abnormal physiology has been seen not only in this disease, but in several of the other cerebellar ataxias. But what we're seeing, and this is not my work, this is Vikram's work now, what he's seeing is that it's different in different diseases. And in some cases it's involving calcium-activated potassium channels. In other cases it may be involving TRP channels. We just don't know. There are some changes in potassium channels.
More question? So I have a general question. When you in your pathways, looks like all the pathways involve the chaperones, but we know that many of the protein which they can fold on their own. So is there any specific difference? I mean, of the role of the chaperone in the protein folding and the misfolding?
Well, I guess let me ask you a question first. How many proteins actually fold on their own? How many good examples are there of proteins that, in the absence of anything else, actually fold correctly at a high probability? I don't know. Right, but how many? I mean,
we don't have that specific data, but when we do the protein folding simulation, for example, we never really, or rarely we consider the help of chaperone. So basically, we suppose a majority of the community, they suppose a protein fold on their own by the sequence itself, from the sequence itself.
yeah, there seems to be a growing literature on co-translational chaperone modulation of protein folding. I don't know whether it applies to particularly big proteins or ones that are multi-domains or what, but there's increasing evidence for that. I don't know, I can't answer the question. whether these proteins are ones which by themselves would be prone to fold correctly on their own.
Okay.
Information on that. I don't know about the Scott program, but I know that in the SPMA, significant, look at PolyQ, Huntington, look at raw protein, all of these are clear H90 substrates. They're clear prion-like, so they have to be, they constantly chaperone, and their activity, whatever it is chaperone by the H90.
Absolutely. I believe there's many of them. There's really folded by the help of the chaperone.
Well,
But
I think it'd be great to answer that question. I'll bet you that over half of all proteins require chaperones to make the right folds in a crowded environment of the cell. That would be my guess. But what? You think it's all, except for ribonuclease A, the original antigen of the experiments, you know. In the crowded environment of the cell, I don't know. That's my guess. That's my hunch. I do believe that these mutations that occur are more likely to make proteins go down off pathway dead ends.
you know, there's a fact is that there's many. Of course, currently there's many computational simulation. Of course, this is a tiny protein, small protein, without any help. They can literally see that in the computer how they fold by itself.
Tiny proteins.
Yes,
You
very
mean
small protein.
like 7 kilodalton or 3 kilodalton? But
Like,
these are mostly 110, 130, 250. It may be that they just all wonderfully fold correctly, but I don't know that.
Yeah, that's a fair question.
Yes.
That ignores the possibility that aggregation happens while it's folding for the simulation.
Yeah. So you mean saying the simulation
The
ignores?
simulation will proceed, but in reality, if you expose a hydrophobic part, you may have aggregation, as mentioned on the.
Right, so when you do the simulations, is it a single polypeptide or is it a polypeptide in a solution? Okay. Okay, that's different. Oh, you're right.
Another kind of question. It has to do with the environment that the brain might be in. Like, for example, some kind of environment that might be related to Alzheimer's, or sorry, related to diabetes, or something that would shift the whole equilibrium. Essentially, I mean, because the brain can exist in different environments, having to do with overall physiological.
Yeah. So so right now in Alzheimer's disease, I'm just going to go off that for a second. In Alzheimer's disease, we know that amyloid is important early in the cascade. And trials thus far of anti-amyloid compounds have been a bust. They have not worked. But they've been tested in people who have fairly advanced disease. So now there are trials being done in people who have identified mutations, but don't yet have any signs of disease. So before the horse is out of the barn, give the treatment. It may work. It may well work. That anti-amyloid therapies are great. But I think everyone recognizes that there's so many other factors that contribute to the brain dysfunction and the brain degeneration beyond amyloid and beyond tau that people are just now starting to look at. Those include inflammatory pathways. Clearly, diabetes is a big deal there. It's one of the hottest areas. And it's what it's actually a key part of our Alzheimer's Center grant is how does diabetes contribute to age-related cognitive decline? Very different from this stuff, but a very important topic.
Okay, if we don't have a question anymore, let's
you.
thank Hank again for the wonderful talk.
Okay, good. I was worried that it wasn't. It's not bioinformatics.
I don't know. I mean, this is about